Hello, everyone. I'm going to introduce an article whose title is DBLiPro, a database for lipids and proteins in human lipid metabolism. Lipids play important roles in cell functions such as energy storage, cell membrane structure, and cellular communication. In the life process, most lipids need to carry out their functions together with proteins. At the same time, the association between lipids and genes is a hot research topic in the field of biology, medicine, chemistry, and material science. With the rapid development of lipidomics, the growing lipidomics data require the development of databases and analytical tools to help researchers work with this data, from lipid identification, pattern recognition, to biological interpretation. The existing popular lipidomics tools are developed for their lipidomics analysis. However, none of the databases offer the ability of multi-omics integrative analysis because of lacking the comprehensive knowledge of lipid metabolism-related proteins. Thus, it is necessary to establish a systematic knowledge base of proteins related to human lipid metabolism, and then construct a lipidome-centric multi-omics integrative analysis framework. In this study, we developed DBLiPro, which is a user-friendly web server allowing for access to human metabolism-related lipids and proteins knowledge. DBLiPro offers analysis workflow for lipidomics, transcriptomics, proteomics, and they are integrating multi-omics analysis focusing on the human lipid metabolism. These are two query examples in DBLiPro. Taking lipid-associated protein O95573 long-chain fatty acid CoA ligase 3 as an example. The relevant gene was identified as ACSL3, and the status was reviewed, and its anomial acid length was 720, and the membrane tags were all true. Taking the core metabolized palmitoyl CoA C00154 related to O95573 as a lipid example. The basic information of its structure, molecular formula, and molecular weights were obtained. The two resources of DBLiPro provide two workflows which have different corresponding input file and diverse output results as shown in figure. The workflow that accepts gene, protein, or metabolized express matrix files has four parts. 1. Data upload. 2. Data preprocessing, including data QC and filtering, missing value imputation, data normalization. 3. Differential analysis, cluster analysis, principal component analysis, and enrichment analysis. 4. Result visualization. The other workflow takes genes, proteins, or metabolized list file as input to select all upregulated or downregulated genes, proteins, or metabolized for enrichment analysis, and visualization according to the user's interactive options. We observed that there are obvious different results according to their different background used in enrichment analysis. This lipid-centered systematic comparative analysis, which could offer unique insights, is mainly thanks to the DBLiPro knowledge base. Through the above analysis, we found that the enrichment pathway focused on the glycerol phospholipids metabolism and the glycerol lipids metabolism. By using the visualization of DBLiPro, the differential expressed lipid-associated proteins and lipid metabolites were integrated together in both glossophospholipid and glycerolipids metabolism pathway. We found that on the overlap proteins of two pathways, diacyl glyosyl kinase epsilon family members were upregulated. By using the data resource of DBLiPro, we found that the reaction between MBO81 and phosphatidylserine based on the core lipids and lipid-associated proteins terms. DBLiPro web server is designed to offer a comprehensive object relational database of lipids and proteins associated to human lipid metabolism and support multi-omics integration analysis focusing on human lipid metabolism. DBLiPro has integrated comprehensive analysis tools for DBLomics, transcriptomics, proteomics data mining. DBLiPro conducts sophisticated and deep domain-centric multi-omics data analysis with biological interpretation and visualization, especially the analysis of lipid-associated proteins and the related functional research. In summary, the new free web server DBLiPro has a comprehensive human metabolism-related lipids and proteins knowledge database. Then, DBLiPro could accelerate lipid metabolism-centered multi-omics analysis. Based on these unique features, DBLiPro could provide insight into understanding the human lipid biological process, which is helpful in the research of biology, medicine, chemistry, and material science.